Hello everyone, my name is Pooja Yadav and today we discussed about the endoplasmic reticulum. Before going in detail with the endoplasmic reticulum, firstly we can see the structure of a cell. This is the cell. The outer membrane is called the plasma membrane that covers the cell. Inside the cell presents cytoplasm and the organelles. The liquid portion is the cytoplasm, and the organelles are nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, secretory vesicles, lysosomes, etc. Endoplasmic reticulum, discovered by Keith Porter in 1945 and George Pallade in 1974. Keith Porter showed through electron micrograph that cells contains a lace-like network of strands throughout the cytoplasm. George Pallade further described the structure of endoplasmic reticulum and Golgi complex. In this, we can talk about the endoplasmic reticulum structure. Firstly, we discussed about the structure, the components of the endoplasmic reticulum, the location of endoplasmic reticulum, and functions of endoplasmic reticulum. So, firstly, in this diagram, we can see this is the nucleus and this is the endoplasmic reticulum. Nucleus contain nuclear pores and nuclear envelope, or we can say the nuclear membrane. The nuclear membrane continues in continuation made endoplasmic reticulum, and in the endoplasmic reticulum we can see the cisternal space, the cisternae, rough endoplasmic reticulum, ribosomes, and the smooth endoplasmic reticulum. Rough endoplasmic reticulum is made up of two layers, or we can say the two membranes, the outer membrane and the inner membrane, and the space between them are called the cisternal spaces. Now, the location of endoplasmic reticulum. All eukaryotic cells have endoplasmic reticulum except red blood cells of mammals. All eukaryotic cells like the muscle cells and the other blood cells like white blood cells, etc. Endoplasmic reticulum is absent in prokaryotes. The entire endoplasmic reticulum is enclosed by continuation membrane. In this diagram, we saw that the endoplasmic reticulum is present in continuation of the nuclear membrane, and the space enclosed by the endoplasmic reticulum, here the space enclosed by the endoplasmic reticulum may represent 10% of the total cell volume. The structure of endoplasmic reticulum is an extensive system of network of membranes, enclosed branching tubules, and flattened sac cisternae that extend from the nuclear membrane throughout the cytoplasm. We can see in this diagram that is an extensive network of membrane enclosed branching tubules and flattened cisternae. Here are the tubules and here are the cisternaes that extend from the nuclear membrane. We talked previously about that line, that nuclear membrane extend throughout the cytoplasm. Nuclear membrane extend and form the rough endoplasmic reticulum, and these are called the cisternae. Then it, in continuation with rough endoplasmic reticulum, made that smooth endoplasmic reticulum, and smooth endoplasmic reticulum is made up of tubules. Okay, now the structure of endoplasmic reticulum. Endoplasmic reticulum extend from nuclear membrane. This is the nucleus. This is the nuclear membrane that covers the nucleus, and it extends form the rough endoplasmic reticulum. And membrane at one end and join plasma membrane at the other end of the endoplasmic reticulum membrane junction. It never open into the cytoplasm. It also continues with other organelles as Golgi apparatus and mitochondrial. We can understand this through the diagram. So this is the nucleus, this is the nuclear membrane. Nuclear membrane continue and form the rough endoplasmic reticulum. Rough endoplasmic continue and form the smooth endoplasmic reticulum, and in continuation with smooth endoplasmic reticulum we saw the Golgi apparatus, then the lysosomes and the plasma, then the plasma membrane. So it never opens into the never open into the cytoplasm, and continue with these organelles continue with the plasma membrane. We can see in this diagram also. See, this is the nucleus, continue with the rough endoplasmic reticulum, then continue with the smooth endoplasmic reticulum, then continue with the Golgi apparatus, then lysosome and secretory vesicle, and then the plasma membrane. So next point, all these structures, tubules and sacs are all held in place by cytoskeleton. The ER remain bounded by phospholipid membrane, and inside the membrane a fluid filled space exists which is known as cisternal space or lumen. This point is understood by this diagram, that the endoplasmic reticulum, roughly diagram, contains two membrane. This is the outer membrane and this is the inner membrane, and these membrane are made up of phospholipids, and inside these membrane a space is called the cisternal space or lumen. See, okay. Now the membrane of ER हमने पढ़ा, the ER endoplasmic reticulum have contain two membranes, outer membrane or inner membrane, and the membranes of ER contain enzymes, triglycerides, phospholipids, cholesterol. Most important endoplasmic enzyme include NADH cytochrome reductase, glucose 6-phosphate, glycosyl transferase, and Mg2 positive activated ATPase. In electron micrograph, the cross section of ER appears as two parallel membrane. We discussed later before, separated by a narrow light space about 4 nanometer wide called cisternae. These enzymes, the role of these enzymes will be discussed in next slides also. So the components of the ER. ER is made up of three components: cisternae, tubule, and the vesicles. Here are the cisternae, vesicles, and tubules. About cisternae. Cisternae, these are long, flattened, sac-like, unbranched tubules arranged parallel forming bundles. Long, flattened, sac-like, unbranched. These are present separated, not in the branch form. Like unbranched tubules arranged parallel forming bundles. Each tubule is 40 to 50 micro thick. Now tubules, there are diverse shape tubules, diverse shape में presents है, and irregular branch 50 to 100 micrometer in diameter. These are found in cells that are active in synthesis of glycerides, and these are also found in big pigmented epithelial cells of the retina involved in metabolism of vitamin A retinol. Vesicles. The structure of vesicles are this. And the vesicles, these are usually round or ovoid structure, 25 to 500 micrometer thick in diameter, surrounded by unit membrane, often occurred isolated in cytoplasm. All these three components are bound by 50 to 60 angstrom thick membrane, and the membrane consists of three layers as plasma membrane, middle thin and the transparent layer of phospholipid, and outer and inner dense layer of protein. Perinuclear space in the is the space present between the endoplasmic reticulum and the nucleus. This is a close relationship between perinuclear space, cisternae, and cell sap. Now we talk about the types of endoplasmic reticulum on the basis of presence or absence of ribosomes on the surface. We divided the endoplasmic reticulum into two, that is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. In this diagram we can see that these are the cisternal like structure form a rough endoplasmic reticulum, and these green structures are the ribosomes, and the presence of ribosomes make rough endoplasmic reticulum, and the absence of ribosomes makes smooth endoplasmic reticulum. Smooth endoplasmic reticulum, absence of ribosomes, and rough endoplasmic reticulum, presence of ribosomes. They can be converted into each other according to their need of the cell. Smooth endoplasmic reticulum, it is highly curved and tubular. They are irregular lattice. Commonly present, the location of smooth endoplasmic reticulum are commonly present in cells connected with lipids and steroid biosynthesis and carbohydrate metabolism. The main function of smooth endoplasmic reticulum are the steroid biosynthesis and the carbohydrate metabolism. The different cells have different amount of smooth endoplasmic reticulum depending upon their functions and nature. Now the rough endoplasmic reticulum is characterized by the presence of ribosomes. Over the surface and connected with the production of secreted vesicles. Rough endoplasmic reticulum is named for its rough appearance, which is due to the ribosome attached to its outer cytoplasmic surface. Rough endoplasmic reticulum lies immediately adjacent to the cell nucleus. Its membrane is continuous with the outer membrane of the nuclear envelope. Rough endoplasmic reticulum is found throughout the cell. Density is here near the nucleus and the Golgi apparatus. In this diagram, what is identify these? So what is this? A is the nucleus. B, what is B? Is the rough endoplasmic reticulum, and what is C is the smooth endoplasmic.